All right, so it's all about exam 3 review today. As per the usual, we can structure it however it works best for you. I've got the PowerPoints from our 3 different chapters loaded up so we can look through those. We've got the exam objectives right there that we can go through and kind of look at the details of the highlighted ones, which are your possible short answer questions. I have some kind of practice questions, practice multiple choice questions that we can take a look at as well to remind us of different topics in different chapters and things like that. And of course, we can, more importantly, address any specific questions that you have. So that may be a good place to start in terms of questions. Are there things that you wanted to review right from the get-go? Things that you wanted to clarify, that you wanted to go through, etc. Yeah, good.
Can we go over number 12? Yeah, 12 and 13.
Of?
Urinary system and anatomy.
So the urinary system exam objectives. Yeah, we can take a look at that. Coming up. So urinary system anatomy, tracings, yeah. So like usual, when we are looking at these systems where we've got a whole bunch of different organs that all contribute to the system and have a different role to play, that's one of the things that we want to review. But then we also, when we have these hollow systems where we have a fluid or a substance that's moving through each of those organs or parts of organs, it's nice to be able to really kind of list out those anatomical structures in order so that we know how things are connected, and then also gets us thinking about, oh, okay, so what happens there, what happens here, what happens with that, etc. So anytime it says be able to trace, in essence, like we did for blood and cardiovascular system and respiratory system previously, you'd be listing those anatomical structures that whatever that substance is would pass by or pass through along the way. So we do have those associated with digestive system, but we had a couple for a urinary system because in the urinary system, we know that inside of the kidney, we've got 2 sets of tubes really, right? So we have a whole series of blood vessels inside of the kidney, both arteries and veins that are routing blood into the kidney and out of the kidney again. Because one of the main roles of the kidney is to condition the blood, to control blood volume and concentration and things like that. So we have to get the blood out to the cortex. That's what we saw originally, is that we see these blood vessels going out to the cortex that then are ultimately going to get from arteries down to arterioles down to capillaries, where we can start doing that kind of treatment of the blood, where we do the filtering, etc. So when we're talking about that filtering then, we're pulling some fluid as well as solutes out of the blood, and now we start running them through a second set of tubes inside of the kidney, not blood vessels, but the tubules of the nephron, and then the parts on the internal anatomy of the kidney where we are collecting this produced filtrate and then urine, and then routing it out of the kidney and then through the other parts of the urinary system. So 2 different sets of tubes that we need to know about, right? So for the first one, if we are talking about our different arteries and veins, in terms of the blood pathway, we'd be using something like this, right? So if we were tracing a, say, molecule of water from the time it enters the kidney until it got filtered out, you'd really be using 1/2, if you will, of what we see laid out in this figure 25.5 of at least this textbook. So we would start out with our renal artery right there. That goes to the segmental artery, interlobar artery, to the arcuate arteries, right? That's where it's curving before we enter into the cortex. Then we get our cortical radiate arteries, which are also called interlobular arteries. You can use interlobular if you want, but just realize interlobular is very similar to interlobar. You don't want to mix them up, right? Cortical radiate arteries get us out to the cortex, where we then slim down from arteries to arterioles, afferent arteriole leads into the glomerulus. And if we were talking about filtering something out, that's where filtration occurs, one of our three processes. So at that point, the water molecule would be leaving the bloodstream. And then the question is, oftentimes in that question it says, well, we got to pass through the wall, this filtration membrane, and what are the structures of the filtration membrane? So that would kind of be the last part of it. When we talk about filtering it out, we have three different parts of the filtration membrane. So those, right, here's the afferent arteriole that we had just listed. There's the glomerulus, and on the filtration membrane, we would be going through, number one, we'd be going through the wall of the glomerular capillary. We'd be going through that endothelial wall. Number two, there's a fused basement membrane in between. So the basement membrane is the middle layer. And then finally, you're going to be passing through what you can either say as the podocytes, or you can talk about how those podocytes are making up what's called the visceral layer of the glomerular capsule, right? So those are the three layers of the filtration membrane. The epithelial wall of the glomerular capillaries, the basement membrane that holds that, glues it basically to the rest of the filtration membrane, which is the visceral layer of the glomerular capsule, or the podocytes. So as it passes through those three things, then it's out of the blood, and now it's on the tubular pathway, but that would be a separate tracing, right? We wouldn't have you do all of the blood vessels going there, and then all of the tubular and filtrate pathway after that. It would be separate. Okay, so mostly your tracing would be this going into the kidney like that. And then once we got to the capillaries, like we said, you would have the parts of the filtration membrane, which is going to look really messy if I write it all out. But we said that there are three parts of it that we had just discussed, right? So you would want to list those, and then that would be the end of your tracing. Okay? If you were talking about somehow the blood that was continuing on going out of the kidney, you would start here at the efferent arteriole, go to the peritubular capillaries instead, and then you're tracing through the veins that have the same name as the arteries that we trace going in. So that would just be the other half if we were talking about blood vessel flow. So either going into the kidney to be filtered out, or the blood that didn't get filtered out as it's exiting the kidney would just be the venous half of that. All right, so that's the blood part of it. If we were talking about instead the filtrate, when the water molecule got filtered out, starting with when it got where it got filtered out, and then tracing it until it exits the body, you'd be starting with the parts of the nephron instead. Okay, so in that case, you could start with. So if it comes out of the glomerulus, what's the thing that's catching that filtrate as it first gets filtered out? Yeah. Yeah, so the glomerulus is the capillaries, and then what's surrounding the glomerulus? What's the coffee pot that we were talking about that catches it? What? Exactly, right? And with the glomerular capsule, don't forget that we talked about two different capsules. There's a renal capsule. That is connective tissue that surrounds the kidney itself, right? There's the capsule, and then there's the cortex region, the medulla region of the kidney. But this is the glomerular capsule instead. So you would start out in this tracing, glomerular capsule, catching that filtrate. From there, what's the first segment that we see in terms of those tubules? What's it called?
PCT.
Yeah. So you would have the proximal convoluted tubule, and you'd want to spell it out just so you know. We know that it's proximal convoluted tubule as opposed to something else. Proximal convoluted tubule. Then we go into that weird loop, right? So we go to the descending limb. Of the nephron loop. Then the ascending limb coming up again. And the reason we're listing those separately is remember that that's another one of our topics that's a little bit tougher, where we talk about that nephron loop and what it does in allowing us to produce concentrated or dilute urine, right? Changing and creating that concentration gradient inside of the kidney tissue. So, and remember that the two sides of the loop, descending and the ascending limb, do different things. The descending limb is reabsorbing water, and the ascending limb is reabsorbing sodium for the most part, right? So they're different. Okay, so then after the loop, we have the distal convoluted tubule. And technically that's the end of the nephron, but where do multiple nephrons dump into? That big tree-like structure. Yep, so you can call it either thing, I don't mind. Collecting duct or collecting tubule. Either one's fine. And then from there, we're actually exiting the tissue of the kidney itself, right? We're exiting the medulla. And now that filtrate, by the time we get to the collecting tubule, which is a little bit, we said, a lot of our convoluted tubules are like the small intestine, where a lot of the work gets done. Collecting tubule is kind of like the large intestine, where it's cleanup crew. It's like, well, do we need to do any more water or electrolytes? Let me know, and I will if we need to, but if we don't, we'll just let it go, right? After we exit the collecting tubule, now it's just urine. It's no longer filtrate where we're like taking things from it, putting things back into it. Now it's urine, and we're done tweaking it. So from the collecting tubule, then we go to the minor calyx, and that drains into the major calyx. From there, that goes into the renal pelvis, which is getting urine from all different aspects of the kidney. The pelvis, what's the tube that drains the kidney? Bless you. Starts with a U. Yep. So we have the ureter. 2 ureters draining our 2 kidneys. That then goes down to the bladder. And then what's the tube that drains the bladder? Also starts with the U. Yep, urethra. Right? So with that tracing, when we're tracing the filtrate and then the urine, the first part of it is going through the nephron, right? So it's microscopic. The second part of it is more macroscopic, right? The big collection areas, the calyces and the pelvis and the ureter and the bladder and the urethra. So this is what you would be doing if you were tracing some filtered water, some water that came out, and now it's part of the filtrate, and it's going to be part of the urine, and where that goes. Okay? All right. Other things. Questions, objectives?
Number 6 on urinary.
6 on urinary? Mm-hmm. Let's take a look at that. So, 6 corticomedullary osmotic gradient. That one? Yeah, that's a big one. That's a big topic. A lot of big words on that as well. All right, so as we were discussing, what's going on there is that's where we're talking about that gradient that occurs from the cortex down into the medulla. And it's the whole role of our nephron loop. Okay? So this is a section, let me see, what slide is that? That's slide 63 in the presentation. So this shows us that gradient, right? So this is in milliosmoles, and it's showing us how in the cortex, it's about the same concentration as what we were filtering out of the blood originally. But as we go deeper and deeper into the medulla, it gets more and more concentrated, up to 4 times as concentrated, right? So there's that overall trend that we're kind of explaining as we look at that nephron. So when we were talking about the nephron, we're doing our filtration here. And now, as the filtrate is going down, right, we can see that on the descending loom, we're reabsorbing water. So as we reabsorb water, we see the concentration of that filtrate increase. When we go up this side, we're reabsorbing sodium and chloride, we're reabsorbing those electrolytes, and that's going to make the concentration of our filtrate now more dilute again. Okay? Now the point is not necessarily the filtrate at this point, but the point is when we're doing that, when we are creating this concentration gradient in the tube, when those substances are transporting out here, it's also creating that gradient in the kidney. So the nephron loop creates that corticomedullary gradient of 300 milliosmoles in the cortex down to 1200 milliosmoles in the medulla. And it creates that so that the collecting duct or the collecting tubule can use it to concentrate the urine as necessary. So what that means is, right, and sorry, before I move to collecting duct, remember that it's the reabsorption of sodium that's occurring on the ascending part that causes the osmotic reabsorption of water on the descending part, okay? Because we're piling all this sodium out here, it creates an osmotic pressure that draws the water out in the descending part, right? Water can only move according to osmosis. There's no active transport of water where it's pushing it one place or another. It only moves via diffusion or osmosis. So we've got to create some sort of gradient for it, right? We have to have a place where water is higher concentration compared to another area where it's lower concentration. And the way that we make a solution with lower concentration of water is we pile more solutes in there. And now, on a relative scale, it's got less water and it's got more solutes. So the water will head into that area to try to balance things out. Okay? So that's the other thing that I want to mention is you have to remember that it's the ascending part, ascending limb, where we reabsorb sodium. And that reabsorption of sodium on the ascending limb drives or causes the reabsorption of water on the descending limb. Okay? So this, as well, one other thing we can kind of throw in there, is that type of water reabsorption that we see there is called obligatory water reabsorption. We talk about that too, and that's another kind of short answer question or other topic. What's the difference between obligatory and facultative water reabsorption? So obligatory, we are obliged to, we have to all the time. Facultative is optional water reabsorption. Do we want it? Do we need to get rid of it? Etc. So we have choices as to whether we reabsorb that water. So the water reabsorption that occurs here in the proximal convoluted tubule right away, and the water that's absorbed here in the descending limb of the loop of Henle, that is all obligatory water reabsorption. We're always reabsorbing as much water as we can in those segments just to make sure that we're not a dried out husk, right? Because we can filter through our 5 liters of blood in totality in short order, right? And there's nothing in the blood vessel in terms of leftover water. So we reabsorb that water in those sections, and then it's only in that collecting tubule, and a little bit of the distal convoluted tubule, depending on what book you read, is where we have the facultative water reabsorption. So facultative water reabsorption, the optional water reabsorption, is under the control of antidiuretic hormone, seen there, highlighted there. So when we have antidiuretic hormone, then the effect of that is we insert those little aquaporins into the wall of the collecting tubule, which allows the water to leave. So the loop of Henle that we were talking about creates that osmotic gradient, creates a very concentrated environment outside of the tubule so that the water is looking over there and says, hey, there's a lot of sodium and things and solutes over there and not a lot of me. So I want to go over there, but that's only possible if antidiuretic hormone inserts those channels, those aquaporins, and provides those little exit points for it to be able to do that and diffuse out. Okay? So, if we need to reabsorb more water, if we're dehydrated and we need to produce a very concentrated urine, we release antidiuretic hormone, which causes the cells in the collecting tubule to insert those little aquaporins, those little channels, and now the water can escape so that we're putting it back into the bloodstream and it's not being lost into the urine, not as much, right? So that is our facultative water reabsorption, and the only way that the tubule, the collecting tubule, is able to do that is that loop of Henle back before has created that concentration gradient, right? So like when we were looking at the water down here, as it's going down the tube, it's going into a more and more concentrated environment, and so more and more water is drawn out of that because of that osmotic gradient. So the tube that we have there, right, in terms of the nephron tube, in terms of that loop, that's what creates the gradient, and the collecting tubule or the collecting duct uses the gradient. Okay? Now, if we, like we said, if we don't need to reabsorb water, if we're not dehydrated and we actually have to get rid of water, then we don't do the antidiuretic hormone, which means those aquaporins are not inserted into the membrane, which means that the water, even though it sees the gradient, can't do anything about it, and the water just exits instead. So that's when we're putting out very dilute urine instead, is when we need to, and we're not having that extra water reabsorption occurring. Okay? In fact, let me, one second, I want to see, there's that animation that I had there that wasn't necessarily embedded, but I wonder whether it'll work in here. This one. Maybe. No? Thought maybe it might work there. There's in essence an animation that shows the difference of that concentration of the filtrate as it moves through the tubule like that. Let's see if I can get it to play over here. Whoopin'. Go ahead, you can whoopin'. It's too old in terms of the technology. Okay. All right, so that still was our answer in terms of that cortical osmotic gradient, cortical medullary osmotic gradient, the fact that it gets more concentrated from the cortex down to the medulla, and how the collecting tubule uses that gradient when we need to allow us to reabsorb water when we're dehydrated. Okay? So for an answer like that, you would want to discuss those two parts. You want to discuss the nephron loop and what's going on in that, and the fact that it creates that gradient with the reabsorption of sodium on one side, reabsorption of water on the other side, and then the collecting tubule uses the gradient. That's what you got to say, uses the gradient, and then you have to explain that it only is able to use the gradient when we have that antidiuretic hormone allowing the insertion of those aquaporins and allowing us to reabsorb more water. Okay? So for an answer for that question, you want to explain those two parts, the nephron loop and the collecting tubule, and then you have to talk about antidiuretic hormone, because that's the only case when that's being released that we can actually use the gradient and reabsorb more of that water.
Just to verify, you said that the collecting tube only uses a gradient when ADH is present, correct?
Yeah. Yep. Otherwise, it's kind of like the windows that we have here, like the water can see, and it's like, oh, I'd love to go that way, but it can't. So then it has no choice, but it basically just goes straight out the tubule, ends up in the filtrate, in the urine. Yeah. Other things. Now remember, we've got the fluid and acid-base chapter, which we just finished. So that's maybe more fresh in your head, but talking about water, how we control water balance in the body, where we find water, talking about specific electrolytes, our sodium and our potassium and our calcium, how those are controlled, and then finally talking about pH in the body and how we have those three different mechanisms that control pH, right? Chemical buffer system, respiratory mechanism, renal mechanism. Earlier than that is the digestive system, where we talk about all those different organs and those different processes, as well as all of those digestive enzymes, right? Question?
Can we do 3 next?
3 of what?
Urinary system.
Yeah, possible. What's that one have to do with?
The nephron.
The nephron itself. Where do I have that? Where did I got that? Here and here and. Is it juxtaglomerular apparatus, that one? No. 3?
Yeah.
Okay. That's a good one, too. So for that one, let's give us some visual references for that. So the first thing is we want to define what the juxtaglomerular apparatus or the juxtaglomerular complex is. You can use either term for it. So the juxtaglomerular apparatus, we actually have to identify that in lab as well, right? So we know on that 3-part model where on the right side we see that zoomed up view, kind of like that, of the glomerulus and the glomerular capsule, we can see it there as well. So the juxta, we can write it out as well, right? So the juxta glomerular, the near the glomerulus apparatus. So sometimes it's called the JGA, sometimes it's called the JGC there. If you call it the juxtaglomerular complex, that equals the juxtaglomerular cells of the afferent arteriole. So that's our inflow, remember? That's an R and an I. Plus the macula densa cells of the distal convoluted tubule. It's kind of right at the junction of the distal convoluted tubule and the ascending limb of the loop of Henle. So we have specialized cells in both cases, macula densa cells and juxtaglomerular cells. Those cells together make up our juxtaglomerular apparatus or juxtaglomerular complex. So we can see them here and here. So together, those things are making up our juxtaglomerular complex or juxtaglomerular apparatus. Okay? So the question is, what is that doing? Why do we have these specialized cells there on the inflow pipe, if you will, and the outflow pipe? Because the distal convoluted tubule is the outflow pipe, right? After we've done a lot of our reabsorption and secretion and stuff like that. So this is that physiological checkpoint where we are checking like the concentration or the osmolality of our filtrate and seeing kind of, oh, how concentrated is it? Is it too dilute? Is it too concentrated? Do we need to tweak that? We're also checking the blood pressure coming in, right? Because the blood pressure coming in is what's driving filtration, which then delivers these things to be reabsorbed, to be secreted, etc. And we said that the movement through our tubules when we're doing our reabsorption secretion is kind of driven by that filtration that occurs originally, because the more filtrate we create, the more stuff is going through the tubules, the more we have to reabsorb, the more we have to secrete. And so we've got to make sure that, kind of like the digestive system again, right, like the small intestine, large intestine, as it marches its way through there, we got to give it time to do its job, to pull things out of it before it gets lost. If it goes too fast through, now we haven't had enough time to recover things from it, right? It shoots through. It's like, oh, we missed it, right? If it goes through too slowly, then we're kind of too efficient at the job. We're pulling too many things out of it, and now it's coming out the other end is super concentrated and maybe not exactly what we want to serve the needs of the body. So that juxtaglomerular complex has mechanoreceptors in it, kind of like mechanoreceptors like pressure receptors, checking blood pressure. Also has chemoreceptors, in essence, that are checking the chemical composition of it. And based on that, if it needs to, we can adjust the glomerular filtration internally to make sure that we are filtering enough or not filtering too much, right? So we know the afferent arteriole is our main inflow pipe. We know that if we adjust the diameter of it, if we constrict it or dilate it, that changes the glomerular filtration pressure. And it's that pressure which causes the filtration. So if we need to kind of slow things down, we can constrict that. And now glomerular filtration pressure goes down, glomerular filtration rate goes down, etc. And if we need to increase that, we can dilate it instead, right? Because it's got that smooth muscle layer to it. So these cells here help to command, if you will, that afferent arteriole and help to regulate the filtration and effectively then the reabsorption and secretion that goes on inside of the nephron. So this is our internal checkpoint, our internal control that we have that can allow the nephron to do its job regardless of kind of changes that are going on outside of the kidney, right? So if there's big changes in blood pressure or something like that, then this can still make its own little tweaks to make sure that we're still doing enough filtration and not too much to make sure the kidneys are doing their job in conditioning the blood, okay? So for the juxtaglomerular complex that we have there, you would need to say that it's made up of those two sets of cells. You need to say that it's kind of an internal, within, right, an intrinsic control point, if you will, that maintains glomerular filtration. So it has, like we said, it has chemoreceptors and pressure receptors in it that allow us to adjust the filtration as necessary so that we can maintain our kidney function. Okay? Now that does also remind us that there's a separate topic, a separate question where it asks about how the kidney and how the activities in the kidney are controlled. That is an intrinsic or an internal control, right? But we always have two types of extrinsic or external control as well. So that's going to be our nervous control and our endocrine control, hormonal control, right? So we see that all the time. We see that for urinary system, and we also see it for digestive system. So that's another like digestive question where it says, okay, so what are the 3 ways and 3 levels of digestive system control? And it's going to be 2 types of extrinsic, neural as well as hormonal, and then there's going to be an intrinsic one as well. So for this one, for urinary system, if we're talking about neural control, it's always going to be, in this case, right, because it's subconscious, it's always going to be autonomic nervous system. It's going to be sympathetic and parasympathetic stimulation that are affecting the activity. Now, in terms of blood vessels, that amounts to just sympathetic. So when we stimulate with sympathetic neurons, our blood vessels constrict, including things like our afferent arterial, etc. So in an extreme case where maybe we are having blood loss or something like that, right? So we have a big cut and we're bleeding out and stuff like that, temporarily we need to shut down what's going on in the kidney. Even though things are going to accumulate, maybe to toxic levels, if we don't maintain our fluids, we're going to die anyway. So extreme level override, sympathetic stimulation, we're going to constrict those different blood vessels, which is really going to bring down our glomerular filtration, like we saw in the 50x exercises, right? We lock it down. So that's a type of extrinsic control, sympathetic stimulation. Hormonal control comes into play when we talk about things like antidiuretic hormone, right? Aldosterone's another one that controls sodium levels. Antidiuretic hormone in particular is effective not only because it allows us to reabsorb water like we saw before, aquaporins, but it also has an effect of being a vasoconstrictor. The other name for antidiuretic hormone is vasopressin. Vaso means blood vessels. So it creates pressure in the blood vessels by causing them to constrict. So if we're dehydrated and our blood volume and our blood pressure is going down, we're doing whatever we can to increase blood pressure. We're holding on to water and we're constricting the blood vessels, and both of those are going to help to maintain blood volume and pressure. Okay? So that's a type of hormonal control. The hormones come from outside of the urinary system, so it's extrinsic, but they have an effect on it. Okay? So antidiuretic hormone's a big one in terms of a hormonal example. So that was kind of, we were adding on to that, right? That's all about intrinsic control and explaining the different cells that are involved and what it does. But part of a bigger question as well in terms of the ways that the kidney is controlled and coordinated. Other things. Yeah. Number 11 on the fluid and acid base chapter, that one? Let's see. 11, explains how and where the following buffer systems work. All right, so that one is a good one because that one really is the chemical buffer system. So that reminds us that for controlling pH, we have 3 different mechanisms. Controlling pH. So we've got the chemical buffers, which basically that's what those are. We've got the respiratory mechanism, which is actions of the lungs that affect pH. And then we've got the renal mechanism, which are actions that are occurring inside of the kidney that also affect pH. Now, when we have those listed like that, so we need to know all three of those, and we need to be able to describe how each one of them works. Be able to give kind of examples and descriptions of how each one of them works. So the question that we are looking at in the exam objectives right now is talking about different examples of chemical buffers. So overall, if we're explaining chemical buffers, those are chemicals, molecules and things like that, that we have inside of the body that can bind up or release hydrogen ions. We know that pH by itself is really focused on the concentration of free hydrogen ions in a fluid, right? And the more free hydrogen ions we have just floating along loose by themselves, the more, is that more basic or more acidic? More acidic. More free hydrogen ions means it's more acidic. On that pH scale, is that the bigger numbers or the smaller numbers? It's the smaller numbers. So the smaller numbers, because it's that dumb calculation, it's not dumb, but sometimes it's hard to remember, that we're looking at the log of 1 over the concentration of the hydrogen ions. So the more hydrogen ions we have, the smaller this number gets. Okay? So that's like 1, 2, 3. That end of the 0 to 14 scale is the acidic end of the scale. Okay. So a buffer is something that is going to, like we said, bind up and hold on to a hydrogen ion, which makes it less acidic, or if we need to, releases it if we're too alkaline or too basic instead. Okay? So different molecules and things like that, or ions that can do that in the body. So that's what we're seeing there. So we see, it says carbonic acid and bicarbonate, hemoglobin, proteins, different types of proteins, and then phosphate as well. Phosphate, PO4, can bind up hydrogen ions. It gives us kind of phosphoric acid. But as long as the hydrogen is bound up in a larger molecule, now it's not contributing in terms of making things acidic. So there are various proteins like that. Of course, hemoglobin is a protein. It's a 4-part protein, as it is in our red blood cells. And when it's not carrying oxygen, if we have a free binding pocket, it can bind up hydrogen ions as well. And so that is also going to change the pH. So those are some examples. And then lastly, the carbonic acid bicarbonate. So that one we usually attach more to the respiratory mechanism. So that one is where we have carbon dioxide plus water gives us temporarily carbonic acid, which then falls apart into a hydrogen ion plus a bicarbonate ion, like that. And that equation can go either way, right? It's a reversible equation. Depending on how much carbon dioxide we have, it can affect that. And I know I just wrote that underneath renal mechanism. But that chemical conversion like that happens in a lot of different places, right? It happens inside of the red blood cells when we're trying to carry carbon dioxide. It happens in the walls of the stomach when we're creating hydrochloric acid. It happens in the walls of the tubules when we are secreting hydrogen ions and reabsorbing bicarbonate ions. Okay? So it happens in a lot of different places, so it kind of counts as our chemical buffers as well. So it counts here. This is something that can bind up and release hydrogen ions, but it also is significant in the respiratory system because the respiratory system is focusing on oxygen and carbon dioxide, and whenever we change the levels of carbon dioxide, it affects this equation. It affects how much hydrogen ions we have floating around. The more carbon dioxide we have, the more it tips this equation that way and converts it to bicarbonate and hydrogen ions, the more acidic that fluid becomes. So if we're not taking care of this carbon dioxide and it's accumulating, our tissues and our fluids are becoming more acidic. On the flip side of the coin, if we are taking deep breaths, regular breaths, and we're getting rid of a lot of that carbon dioxide, it goes the other way. We combine the bicarbonate ion and the hydrogen ion to release water and carbon dioxide that we're breathing off. So it goes that way instead. So it makes us more alkaline or basic. Okay? So this explains our respiratory mechanism. It kind of focuses on carbon dioxide, but it has an effect on pH. But it also kind of counts as our chemical buffer because we use this in various different tissues around the body as well. Okay? So that's why we have it listed there. So for each of these, so that transitions us to that. If we're talking about controlling pH, you want to be able to talk about each one of these and how they work. So be able to say that these are chemicals, right? Molecules or ions that are in a solution that can bind up or release hydrogen ions, right? It prevents really big changes in pH from occurring because we can take care of those almost immediately. So chemical buffers are really fast because those ions and molecules are just there and ready to bind up free hydrogen as necessary. Respiratory mechanism focuses mainly on oxygen and carbon dioxide, but when we change those levels of carbon dioxide, it affects pH. So when we're breathing, it also affects pH. And then the renal mechanism, that comes into play because, like we talked about, we can secrete hydrogen ions. It's one of the two examples of things that we commonly secrete, hydrogen ions and potassium ions. And so when we do that, we use this equation again. We get rid of the hydrogen ions, we reclaim our bicarbonate ions, and that also affects pH. So technically, even when we're acidic or basic, we can hold on to hydrogen ions if we need to, even in the kidneys. So, but that takes longer, right? It takes longer for everything, the blood to flow through and to go through all the different tubules and stuff like that. So this is also kind of the timeline in terms of these buffer systems, in terms of how they work, right? This is almost immediate. This is minutes to hours in terms of changing your breathing and having it affect pH. This is more like hours to days as the kidneys are continually working on blood. Okay? Okay, very good. Other things. Now remember, as we're going through these, you got to practice with these too, right? You got to practice with these in the next couple of days. You got to write this stuff out yourself, get your own answer, and then practice writing it out, right? So not just like laying it out like I have the answer, it's right there, I'm just going to read it. You got to be able to like hand that card to somebody else or flip that card over and hit all those different points. And keep doing it until you hit all those points. So you have a solid answer, not just like, well, I named them, but I couldn't remember the examples or how they work. That's only going to give you partial credit. You don't want that. You want full credit, especially with these types of questions where we've got a clear list of what's going to be expected. Just rise up to put the time in so you can meet those expectations. It's more of the multiple choice section where you're like, I don't know exactly what's going to be there. That's why I'm reviewing everything, right, that we've been talking about. But for the vocabulary and the short answer, you know where it's going to go. So right now, we're kind of getting all of that in order so that we know what your answer is going to look like. But this is the time where you really want to practice with that. Test yourself. Have other people test you. Yeah?
Should we start going through the multiple choice questions?
Yeah. I think that's a good idea. That way we can kind of hit some different topics that maybe we haven't been thinking of as of yet. So let's take a look. I think I had those there, and then I folded them up here. We got just a few going through different questions having to do with our different systems. Okay, so here's one. This one comes from our fluid and acid-base chapter, our last one. And this one is addressing, water balance, where do we keep it, and how do we control it, and stuff like that. So sources of water gain include. So we need to know how we gain water, we need to know how we lose water, and it's keeping that in balance. What do you think? D is in dog?
Yeah.
Is that what you said? Okay. So ingestion, that's a good, I don't know why it says ingestions, but okay. Ingestion, that's one of our 7 different digestive processes. That's taking things into the mouth, right? That's a good review for that. We have to know those 7 processes and give examples about where they occur along the digestive system. Ingestion is just in the mouth. Secretion is all along the way in terms of mucus and different glands and stuff like that. Propulsion, chemical digestion, etc. But ingestion of food, for sure, there's water in our food, different values. There's the most contribution of fluid or water intake is going to be from ingestion of liquids. How about that last one that we see there? It's possible, right? We said that there is metabolic water that contributes a small percentage in terms of our balance like that. So metabolic water, when we're talking about like dehydration synthesis and things like that, but also water is part of our process as we're doing even cellular respiration as well in terms of aerobic, because we need oxygen as part of it. And so that comes into play along with that as well. So there are some chemical reactions that affect that water balance, that at the end of those chemical reactions, as a byproduct, we've either made water or water has gone into making that chemical reaction occur. So we got to remember that as well. So we usually are just calling those, like we said, metabolic water. Okay, I threw this slide up there as well, though, because when we're talking about water and water balance, we also need to know the major places where it's stored, right? So there's more intracellular fluid than extracellular fluid. And then when we're talking about extracellular fluid, because that's just one category inside of cells, but extracellular fluid, the largest category that we have for that is interstitial fluid. That's the fluid surrounding the cells in our tissues. Then we have things like plasma and synovial fluid and the humors of the eye and things like that, right? Cerebrospinal fluid, etc. And then finally, remember when we were talking about water, we needed to talk about, again, different hormones or different control mechanisms. That's mainly going to be our antidiuretic hormone, right? That causes us to retain water. Interestingly enough, we said that allows us to reabsorb water in the kidney, works as a vasoconstrictor, and also is a thirst stimulator in the hypothalamus. So the hypothalamus is not only in control of like food intake and things like that, hunger and satiation, but it's also our thirst center. So when antidiuretic hormone is released, it, because blood flow, right, ends up in the brain as well, and it can stimulate us to drink more as well. So that's going to influence the other side of that equation, our water intake as well as our water output, right? So it has a lot of different effects on water. The other thing that we talked about is not only where is water found in the body in different compartments, but also these electrolytes. So this one was a good slide, I always feel, because it graphically, as a picture, shows you the major electrolytes and where they're found. So on the left side is where we see, again, our ECF, and on the right side, we're seeing an example of an intracellular environment, right? And we can see the concentration is different. So we see sodium out here. There's a lot of calcium out here in the extracellular environment as well. When we look at the intracellular environment, it's opposite, right? There's not very much sodium, but there's a lot of potassium. There's our phosphate. We said phosphate's important in terms of binding to hydrogen ions. There's a lot of different proteins in there as well that can affect pH, that have other jobs to do and contribute to that negatively charged internal environment. Okay? Okay. Look at this one. Digestive system, what that is, right? So we talked about different digestive organs and different aspects about them. We had to know parts of the teeth and, different types of salivary glands and different regions of the stomach and layers and stuff like that. So this dental formula that goes 2, 1, 2, 3 reminds us of our different tooth types. And each type contributes a little bit, right? Biting pieces off, grinding things, etc. So when we're talking about the one that we see here in this 2, 1, 2, 3, what type of tooth is that referencing? Right? So we've got 2 incisors. There's a medial and lateral incisor. There's 1 canine. There's 2 premolars, and then there are 3 molars. That's starting at the midline and then going laterally, going to the side, whether you're on the top or whether you're on the bottom, right? So it just kind of reminds us that way. So that's 8 different teeth times 4 gives us our 32 adult or secondary teeth, right? Okay. How about this one? So this takes us over to urinary system instead. The last one was a little bit easier, hopefully. This one's a little bit more complex. Not only because it has a bunch of words to it, but it's also referencing this stuff that's called renin. So what do you think? So I can tell you that this renin is part of the RAS mechanism. So the RAS mechanism starts out with renin being released actually from the juxtaglomerular apparatus, interestingly enough. So it kind of ties together our internal and our external controls. This is a kind of a chemical domino effect. The renin that's released causes angiotensin to be created and released, both 1 and 2. And then that causes the release of aldosterone. So it's the renin, angiotensin, aldosterone system. It causes the release of aldosterone. So that's the link that we have there for renin. So we said insufficient quantities of renin. So according to that domino effect, that means less angiotensin. That means less aldosterone. Now the question is, what does aldosterone do? The final step in that whole domino effect there. Yeah. So it causes the reabsorption of sodium in the kidney. It's one of our 2 hormones, like we did in the PhysioX exercise, that affects the concentration of the urine, right? Now that one's targeting sodium and potassium. So it allows us to reabsorb sodium, which allows us to reabsorb a little bit more water because of osmotic gradient, and causes us to secrete potassium as well. So then, given that, if we look at those answers, the patient will not be able to activate enough angiotensin 2. True, right? Because without the renin as the first domino, we're not going to get angiotensin, we're not going to get angiotensin 2. Okay? B, the patient will not be able to release enough aldosterone. That's the 3rd step, technically the 4th step, right? So if we don't have enough angiotensin, or 1 and 2, then we don't get aldosterone. Okay? So right off the bat there, if you can figure that out, you've got 2 that you're confident in knowing that they're true. You got to go with all of the above. Right? If you're confident that you have 2 out of those answers, you got to go with all of the above. You definitely check out those other answers to reinforce it and say, yep, that one's I think is true as well. But if at least you have 2 that you're confident in, then, and there's that extra choice, you got to go with that one. So last one, we said sodium and potassium are going to be affected directly. pH balance can be affected as well because, in fact, those sodium-potassium pumps can be sodium-hydrogen pumps. So when we are reabsorbing sodium, we actually kick out hydrogen. So it affects pH as well, or it can. Last one says the patient may have difficulty maintaining blood volume and therefore blood pressure. Yeah, that's because, again, even though we're reabsorbing sodium, water, as the little brother, tends to follow it. So that affects our concentration and volume of the blood. So it can have an effect on that blood volume as well. So that one was talking about some of our internal controls as well as external controls. So that references our juxtaglomerular apparatus again, because that's where our renin comes from originally. Okay? All right, how about this one? Still in the kidneys. We're in the nephron itself, and this says, as tubular flow, or fluid, flows up the ascending loop of Henle, what happens? Yeah. C, concentration decreases. And on the one hand, you're looking at that, you're like, I don't know if that seems right? We're always talking about how the kidneys are able to concentrate the urine, etc. But you got to think the other way too, that we need to be able to produce very dilute urine as necessary. And so the loop, the one thing we focused on with that darn nephron loop is that it creates a concentration gradient. So that as you're going down one side of the loop, it's starting out dilute and it's becoming concentrated, but it's a loop, so it comes back up the other side. And as it goes back up the other side, now it is becoming less concentrated. So on the ascending side of the loop of Henle, we see that it's becoming less concentrated, right? Just like the surrounding tissue is becoming less concentrated as we go up towards the cortex. But it's not about the concentration in the filtrate as of yet. It's about creating this concentration gradient out here that can be used in the collecting tubule or the collecting duct if we need to, right? So that question itself addresses a couple things. Number one, it addresses what's going on in that loop. Why is the loop there? And why it seems so weird that the filtrate inside of it becomes more concentrated as we go down the descending part, and becomes less concentrated going up the ascending part. And that's because it's not, the concentration in the filtrate at that point is not that important. The concentration in the surrounding tissue as it goes from cortex to medulla is important. It's establishing that. So, concentration remains unchanged. That's not true. We need to create that concentration gradient. We have to, those next few answers are referencing, okay, let me think about what's actually going on between those two sides of the nephron loop, right? On the descending part, we're reabsorbing water. We're pulling water out. That means the filtrate is getting more and more concentrated, right? Because water is being removed. It's now leaving the solutes behind. Now we're going the ascending part. What's going on in the ascending part? We're reabsorbing what? Sodium. So we're pulling our solutes out, so that's making that filtrate more and more dilute, right? So it's diluting it again. We're pulling out our electrolytes that make it concentrated. And then these two here, these are hormones, right? We recognize those as really important hormones in terms of kidney function, but those are only occurring after the loop, right? Like in the distal convoluted tubule and the collecting tubule. That's the last segment where we're like, hey, what do we need to do with this? Are we done with this? Are we good? So we're checking to see, do we need to reabsorb more sodium? Do we need to reabsorb any more water? And so that type of hormonally controlled reabsorption is occurring after the loop instead. Okay? All right. Let's look at, I put that one in there because there's a couple figures that I usually put at the end of the lecture that does a good job, I think, of reminding you about what's going on in each of the different sections of the nephron and the collecting tubule. Okay? Because we said, bless you, that there are three different processes occurring. There's filtration, there's reabsorption, and secretion. For each of those, we've got to be able to explain what it is, give a definition for it, what the force is behind it, right? So like filtration, it's blood pressure. That's what's pushing these things through the filter, is blood pressure, right? For something like reabsorption, the force behind it or the mechanism is like facilitated diffusion or active transport or something like that, right? That's the way that we're grabbing these things and putting them back into the blood. Osmosis, that's another way that we're reabsorbing things specific to water, right? Those are the specific mechanisms or forces that are pulling these things back in. And then secretion is kind of the other way. That's usually active transport, where we're pushing them out and we're using energy to do so. But then this figure shows us different examples, because we need examples of, okay, what things are filtered, what drops through the filter, what stays in, what things are reabsorbed, what things are secreted. We need to have examples for all of those. And so in these figures, wherever you see an arrow that is leaving the tubule, it's leaving the tubule and it's going to be picked up by the blood, right? So going from the tubule to the capillaries, that is reabsorption. If you see an arrow going the other way, going into the tubule, that's going from the capillaries into the tubule, that's secretion. Okay? So this gives you some examples of different things that are secreted, different things that are reabsorbed, and where that occurs. Okay? Because it's a little bit different. Okay. So make sure you review that. So, this addresses that, right? So reabsorption goes from where to where again? So it's into the capillaries from the tubules. It's going from the tubules where, if we left it in there, it'd be lost. We'd pee it out. It's going from the tubules, we're putting it back into the bloodstream so that we can keep it in the body, right? So reabsorption here of glucose and amino acids, how is that happening? So with that one, the concentration is actually fairly high in the filtrate, right? Because we're filtering it out. It's a small molecule and it's being filtered out, so it's fairly high. So in this case, it's kind of like a facilitated diffusion. It can't get across the membrane, we need to assist it. You might even say, it's almost like a hybrid answer here, I would say, because oftentimes when we're reabsorbing it, it's like it's a transport molecule, but it's like a secondary transport. So we're like reabsorbing sodium, and we're expending energy to do that, and then glucose kind of hitchhikes and comes in through that same channel. So, but it's definitely not just regular diffusion, definitely not osmosis. Pretty sure it's not pixie dust. Each of the following can ordinarily cross the filtration membrane except. Of those things, what is going to be able to get through the filter? What's the one thing that can't get through the filter in the glomerulus? What's the biggest thing that we have in that list? Platelets, right? Cells and cellular elements, those are huge. We can actually see those in the microscope. You cannot see molecules, you cannot see ions in the microscope, right? Those are small things that are going to drop through the filter. So small proteins, big proteins, no, they're too big. Small proteins, yep, they can get through. Amino acids can get through, monosaccharides, right? So our simple sugars get through, ions, water. We know all that stuff can drop through the filter. Because the filter, the glomerular capillaries in that filtration membrane, it's size selective. It's not good and bad. It's not do we want it, do we want to get rid of it. It's small, it gets through. Big, it stays in. Okay? Oh, how about this one? Enteric nervous system. Everybody's like, I don't even remember. Did we ever talk about that? So enteric nervous system, GI, so that's gastrointestinal. So this is back for digestive system. The enteric nervous system, we said there's 3 levels of control with the digestive system. It can be sympathetic and parasympathetic, that's autonomic. Sympathetic is going to slow it down when you're stressed. Your digestive organs are all going to slow down. When you're resting and digesting, like next Thursday, then autonomic, in terms of parasympathetic, is going to be activating our digestive system, right? When you lay down on the couch in your stretchy pants. Yeah. So we have autonomic control, we have nervous control, we have hormonal control. Do remember that we had one slide where we're like, oh, what are the major hormones of the digestive system? We talked about gastrin, we talked about secretin, and we talked about CCK, cholecystokinin. We need to know those 3 hormones as examples of hormonal control and what they do. And then finally, this is our intrinsic control. This is our local control, the enteric nervous system. So enteric nervous system, what do we think in terms of an answer for this one? There's too many choices, huh? Yeah, we don't like that. Yeah. I could go for an E on this one. So one of the things to remember with our enteric nervous system is that it controls muscular activity because it sends neurons to our longitudinal and circular muscle, as well as oblique muscle in the stomach if we're talking about that. And that's part of our myenteric plexus, and there's a submucosal plexus. In the submucosa, in the wall of our gastrointestinal tract, that's where we have a lot of our glands, right? And so we can control their secretion as necessary with neurons going to that level too. So this is kind of how it controls the muscular activity, the physical activity, as well as the secretion. But the enteric nervous system, more importantly, is communicating between different organs, like communicating between the duodenum back to the stomach. So they can tell the stomach, yep, you need to slow down because I have a full load of food you just sent me. So I need you to stop churning, I need you to stop secreting, I need you to slow it down, and then I'll let you know when I'm ready. And then I'll send you another message, and now it says, okay, now kick up your churning again, your muscular contraction. Kick up your secretion again, and I'm ready for more. Okay? So enteric nervous system is our internal, intrinsic system that helps to control and coordinate between the different organs of our GI tract. Okay? All right, how about this? We talked about the GI tract, the main pathway, but then we also know that there are accessory organs of digestive system. We need to know each of them and what they do and how they contribute. So when you look at this list, there should be something in there that does not contribute digestively. What do we think?
Would
B, the appendix. That is true. So the appendix hangs off of the digestive system. Where does it hang off of? What's the specific pocket thing that it hangs off of? The cecum, right? So it's along that pathway, but its main role is kind of immune system instead, right? Bodily defenses. It commonly houses a lot of different types of defensive cells, white blood cells, but it also helps to house beneficial symbiotic bacteria in there to help maintain that population of symbiotic bacteria that line the colon or the large intestine. Okay? But there's nothing digestive. There's no food that goes into it and digests. It doesn't produce a secretion that jumps in like the pancreas or the gallbladder or anything like that. Okay, then finally, just in terms of this one, I was using it as an example to remind us of not only our major organs, but also the processes that occur along our digestive system, right? So we've got to be able to say, what is ingestion, mechanical digestion, chemical, propulsion, absorption, defecation, and the other one is secretion. Exactly what is it and where does it occur? Because some things only happen at certain places, like ingestion only in the mouth, defecation only at the anus. But secretion happens everywhere, right? Stomach secretes things, pancreas secretes things, intestine secretes things, like in our duodenal glands, stuff like that. But we always have mucus secretions all along our internal epithelial border as well, so that counts as secretion. So make sure that you know those different processes and where those occur. One other thing in terms of digestive system that we would want to review is those digestive enzymes, right? So that's a lot of the final processes that occur in the digestive system as well. So if we go down, way down to the end of that lecture, we had a section where we were kind of reviewing what was going on in terms of those food molecules, right? So you need to know that there's 4 major nutrient molecules that we're looking to break down in our diet, right? So we've got carbohydrates, we've got proteins, we've got lipids, and we have nucleic acids. Those, you need to know those 4, okay? And each time we talk about any of those 4, we have to say, okay, what's the big form? What's the macro molecule that we're looking to break down? What's the product? What's the smaller form that we get after we physically and chemically break it down? What's the enzyme that does that? Because enzymes are very, very specific, right? You can't throw lipase at a protein and expect it to work. It's not going to. It doesn't fit, right? So what's the enzyme that does that chemical digestion and starts snipping those bonds? And where does that enzyme come from as well, right? Because we know that there's only certain glands along the way or certain sections along the way that actually release enzymes that mix with the food. And then finally, once we break it down small enough, the whole idea is to break it down small enough so we can absorb it across the body wall, put it into the blood, and give it to the cells around the body. How do we actually absorb it across the body wall? How do we absorb it across the intestinal wall? What's the mechanism? Is it osmosis? Is it diffusion? Is it active transport? Things like that. Okay? So for each of those, you want to have examples. So like we see, you can make like a flashcard for each one of those. Carbohydrates, write that on the front. And then, like we said, what's the big molecule? What's the small molecule, etc. So we've got, remember, polysaccharides. That's going to be the large molecule, our large complex carbohydrates. So polysaccharides go down to like di and monosaccharides, our simple sugars. For proteins are actually the large form, right? That's as big as it gets. Those break down into peptides, shorter chains, right? And finally break down into amino acids. For lipids, now lipids are a little bit tougher in various ways, you'll remember. Hopefully, if you don't remember now, in the next couple days you'll remember, okay? They include a few different things, like cholesterol and like fats and triglycerides and stuff like that. So it's kind of a bigger category, but we keep using the example of triglycerides as our main type of lipid, glycerides, So triglycerides, 3 parts, right? Break down into monoglycerides and fatty acids. Remember that from our PhysioX exercise, right? So that's for our lipids. And then finally we have nucleic acids like RNA and DNA. And those break down into our nucleotides. Okay? So we all know what's the big thing that we're trying to break down. Give an example of an enzyme that does that, and then what are the products that we get at the end. So for like polysaccharides, one of our main ones that we were seeing is amylase, right? Amylase breaks down amylose. Amylose is a big starch, okay? So then the question is, where does amylase come from? Well, there's a salivary amylase. You could use that as an example. There's a pancreatic amylase, comes from the pancreas. Pancreas has enzymes for everything. Then we do have these brush border enzymes. Remember, brush border is referring to the microvilli that look like a brush in the small intestine. So right at the very end there, as we're bumbling along in the small intestine, there are enzymes that are embedded in those microvilli that finish kind of cutting those molecules into their smallest parts. Okay? For proteins, where is our first enzyme for proteins come in? So our first enzyme for proteins is actually pepsin, or pepsinogen originally. Where is that released? In the stomach, right? Pepsin or pepsinogen. Pepsinogen is the inactive form. Pepsin, when it's in the presence of hydrochloric acid, is now the active form. That's where we first start chopping up these proteins into peptides. Then, by the time we get to the pancreas, pancreas has a whole bunch more of those, right? So we have different proteolytic enzymes that we have in the pancreas dumping in to mix with the food in the duodenum. And then, again, we have more brush border enzymes, last ones at the end. So those are some examples of enzymes for proteins. In terms of, absorbing across the body wall, right? So this is just showing us some of those examples. So we've got like a type of facilitated diffusion as we're kind of pulling things in. It can be an active transport, it's possible. So you can see, as we're talking about, there's our pepsin, as we're talking about proteins and pancreatic enzymes and things like that. Then we get to our lipids. This one's a little bit more complicated, like we mentioned. So in this case, remember, one of the things that we talked about is that these fats need to be emulsified. Emulsification comes from our bile salts, which is in the bile coming from the liver and the gallbladder. Okay, so that's not an enzyme, right? We talked about that in PhysioX. We said that's mechanical digestion. It's physically separating it from bigger things to smaller things, right? The enzyme comes into play when we're talking about things like lipase. So the lipase comes in. We have lingual lipase, that counts. There's a gastric lipase technically for like milk fats and stuff like that. But mainly, digestive system, we're looking at pancreas, pancreatic lipase, and then continuing on from there. So when we're absorbing that, that's a little bit different. We have these me cells that we package it into to transport it to the walls of the intestine. And then coming out the other side, we package it into these chylomicrons to have it exit the cells lining the intestinal wall and put it into the lacteal, right? Most of our other digestive products that we were talking about before, those go straight into capillaries that are in all of our villi, right underneath the wall of the intestine. But these packages, these chylomicrons, they're too big. We can't jam them into a cardiovascular capillary. So that's why we have those lacteals. The lacteals can handle that. Okay? So like we said, for our lipids, like our triglycerides, it's a little bit more complex. We still need to know the enzymes, but now it's like, don't forget about the bile salts. And when we're absorbing it, it's about those micelles putting it into the cell, and the chylomicrons putting it into the lacteal. Okay? And then nucleic acids, those enzymes are mainly from the pancreas, pancreatic enzymes for ribonucleases and deoxyribonucleases. So that is a key part, to be sure, of the digestive system as well. So not only big picture stuff, what are the organs, stuff like that, but enzymes are really important. So make a flashcard for each of these, and list that critical information on the back, because that's going to come into play somewhere. It's going to come into play multiple choice question, maybe get a short answer question, etc. Okay? All right. So hopefully that helps. Now's your time, though, to really focus on some of that content in preparation for our exam. We have our lab quiz, or not our lab quiz, our lecture quiz we do tonight, right? Lecture exam due on Saturday. You can do it early if you want. It's up to you. But due by midnight on Saturday. Got office hours today. If you want to come by, you can always email me with questions as well.